Good evening, ladies and gentlemen. We are Group 3 of DBM3B, and we're here to present to you how one can distinguish basic features of bacterial culture on agar plates, agar slants, and broth, as well as to recognize the advantages and limitations of cultural characteristics in the identification of bacterial species. Allow me to introduce to you my fellow colleagues who will be explaining their parts of this video presentation. We have Ms. Meiko Lugtu to explain characterization of bacterial culture in agar plates. We have Ms. Sara Lakay and Mr. Juan Gutierrez to explain characterization of bacterial culture in agar slants. And lastly, we have Mr. Sir Gabriel Tener to explain characterization of bacterial culture in nutrient broth. Let us now begin the presentation with Ms. Meiko.
Different types of bacteria will produce different looking colonies. Some colonies may be colored, some colonies are circular in shape, and others are irregular. The characteristic of a colony shape, size, pigmentation are termed the colony morphology. These characters used to accurately describe the morphology of a bacterial colony. Now, how would you describe a colony? A colony is defined as a visible mass of microorganisms all originating from a single mother cell. Therefore, a colony constitutes a clone of bacteria all genetically alike. Different species of bacteria can produce very different colonies. Now let's proceed. These are the characterization of bacterial culture in agar plates. First is the pigmentation of the colonies or color. There are three ways a colony may be pigmented. First is non-pigmented colonies may be described as colorless, white, or off-white. Second, non-diffusible pigment means that just the cell is pigmented. And third, diffusible or water-soluble pigments will color the agar around the colony. Most bacterial colonies appear white, cream, or yellow in color. Now, let's take a look at some of the live pictures of the colonies with different colors. As you can see, we have here the black bacterial colonies, yellow bacterial colonies, orange bacterial colonies, white bacterial colonies, brown bacterial colonies, and red bacterial colonies. Bacterial colonies also has surface characteristics. Dry or powdery, it may be smooth or glistening, wrinkled or rugose, mucoid, contoured, irregular, smoothly undulating, radiate, ridges radiating from the center like spokes in a wheel, and lastly, concentric. It is marked with rings having a common center. Now let's talk about different shape of bacterial colonies. First is punctiform. Punctiform colonies are distinguished from circular colonies by their very small size. Circular, any round colony regardless of type margin. Filamentous, a colony composed of long, interwoven, irregularly disposed threads. Rhizoid or root-like, it is a branching structure. Spindle, which are lens-shaped, and lastly, irregular, not circular, and may be spread rapidly over a plate. Let's take a look at some of the live pictures of colonies with different shapes. We have here, the first picture is circular, the second one is irregular in shape, and lastly, it is a punctiform. Now let's proceed in the size of colonies, preferably in millimeter. Colony size depends on the growth medium and the number of colonies present on a plate. The size of the colony can be a useful characteristic for identification. The diameter of a representative colony may be measured in millimeters. Tiny colonies are referred to as punctiform. Punctiform is less than 1 millimeter, small 1 to 2 millimeter, medium is 3 to 4 millimeter, and large, which is 5 millimeter. Now, take note that colonies larger than 5 millimeter are likely to be motile organism. Next is margin. It describes the shape of the edge of the colony when magnified. The margin or edge of a colony or any growth may be an important characteristic in identifying an organism. Several examples are shown below. Number 1, entire, it is smooth and does not have notches. Second is undulate, it is wavy with shallow indentations. Erase or serrate, it is irregularly notched. Fourth is filamentous, long, irregularly placed or interwoven threads. Fifth is curled, parallel chains in wavy strands. And lastly, lobate with wavy and with deeper indentations. Lastly, odor. Let's take a look at some of the live pictures of colonies with different odor. First one is Pseudomonas aeruginosa, fruity grape-like scent or smells like taco chips or tortilla chips to some. Common cause of burn and wound infections, UTIs associated with formation of kidney stones, causative agent of otitis externa or swimmer's ear. Common cause of skin infection associated with contaminated whirlpool tubs and saunas. Next is Streptococcus aeruginosus, characteristic, sweet, cake-like, caramel or butterscotch odor. Part of the normal skin flora but can cause rare infections such as brain abscesses, liver abscesses, and bacteremia, usually resistant to bacitracin and susceptible to penicillin. Third one is the Escherichia coli. It is floral, flowery scent, lactose fermenting, gram-negative bacilli, part of the Enterobacteriaceae family, may be mucoid or non-mucoid, most common cause of UTIs, and may cause other types of infections such as wound infections or peritonitis, sepsis, and bacteremia. And then we have Echinella corrodens, produces a characteristic bleach smell on blood agar. It is a small gram-negative bacilli or coccobacilli that is associated with human bite wounds. Lastly, Proteus mirabilis smells like rotten chicken, chicken soup or broth, associated with UTIs, burn and wound infections, outer ear infections, and formation of kidney stones.
Next, we have Miss Sarah and Mr. Juan Rafael.
Characterization of bacterial culture in agar slants. Agar is a substance obtained from red algae which is commonly used for growing bacteria cultures. To promote bacterial growth of specific species of bacteria, various substances are mixed. For example, Staphylococcus bacteria growth is favored by mannitol salt sugar which contains sodium chloride and can determine which are pathogenic or non-pathogenic strains. Slanting. Agar slants are created by boiling the agar until it reaches its boiling point and pouring it into a test tube. Test tube is placed in a slanting position before it solidifies. It can be positioned upright if it is already cooled and the agar is in slanted angle. The advantage of agar slants. Slanting the agar in a tube is creating a larger surface area for the bacteria to grow. Water loss is minimized when test tubes are used, and also it can be covered or capped, which is important due to the high moisture of culture media. Uses. Used to culture bacterial cells for the purpose of identification. Bacterial cells will quickly divide, and after several hours, it will produce enough cells to be examined microscopically. It is also useful in maintaining bacterial cultures than a pile of petri dishes. It is easily placed in test tube racks and stored in the refrigerator. First characteristics is the amount of growth. The abundance of bacterial growth can vary depending on. First is none. No presence of bacterial growth in the slant. It can be an indicator that there is an error during the process of the experiment. Next is scanty. There is a growth of bacteria, but its quantity is small or insufficient. Next, moderate. Half of the tube is covered with bacteria or average in amount. And lastly, abundant. Plenty of bacteria grows in the slant medium. Here are the example of the amount of growth of bacteria in a agar slant. In scanty, we have Micrococcus colonies. In moderate, Micrococcus colony. And in abundant, Sporothrax scanty.
So next characteristic is the pigmentation or color of the colony. Some bacteria produce pigments which are colors that they produce. They produce this pigment for many different reasons and functions. They can serve as a protection to the bacteria, aid in photosynthesis for the bacteria that are photosynthetic. Example is cyanobacteria. Also, it can be antioxidants and byproducts of their metabolisms. Some produces these byproducts to protect them from solar radiation and etc. So this pigment and the intensity of the pigment can change based on the environmental conditions. For example, some bacteria produces a certain color at a certain temperature, and at another temperature we can see another color. So we have first color is the reddish or pinkish. We have here Micrococcus roseus, which has rosy pink color at both temperature of 22 degrees Celsius and 37 degrees Celsius. It has a more intense color at 37 degrees Celsius because at higher temperature, the bacteria produces a more intense pigment. The second picture shows here is the Serratia bacteria, which at 22 degrees Celsius and lower temperature, it is a reddish color, and at 37 degrees Celsius, it has a clear white color as we can see on the picture. Next is the Bacillus globuli in the center. This shows there is a two different temperature range at different temperature of 22 degrees Celsius and 37 degrees Celsius. At 22 degrees Celsius, it is orange, and at 37 degrees Celsius, it is much intense pigment like dark orange to reddish brown. Next. So, and the second color is blue. We have an example here, which is the Bacterium violaceum, which is grown at 22 degrees Celsius and 37 degrees Celsius. This bacteria produces a blue-violet pigment. Next is the second pictures showing Gentinobacterium lividum, which is blue to violet color at 22 degrees Celsius and does not grow at 37 degrees Celsius as we can see on the picture. So next, the next color is yellow. So here we have the example of Micrococcus luteus, which is at 22 degrees Celsius and 37 degrees Celsius. It grows out at both temperatures and looks the yellowish color pigment. And lastly, we have the green color. Here is the example is Pseudomonas aeruginosa, which is associated with different infections. It has a yellowish pigment, which is grown at 22 degrees Celsius and 37 degrees Celsius. It has a strong green color at 37 degrees Celsius, and this is significant because this helps us distinguish Pseudomonas from other bacteria. So pigmentation and coloration of different culture is important for us to know the pigment of bacteria because it helps us distinguish specific bacterial cultures. Next is the consistency of bacterial growth. So consistency of bacterial growth in a nutrient agar slant may vary and be classified as the following descriptions. First is butterous or butter-like consistency. Second is viscid, which is it grows, it growth follows the needle when withdrawn. And next, thirdly is the membranous, which appears like thin, coherent like membrane. And lastly, for consistency, we have brittle, which appears like dry and friable. So next is so next slide, this is the example of butyrus consistency, viscid consistency, and brittle consistency. Next. Next is the optical characteristic of bacterial agar glands, which can be described as the following. First is the opaque. It does not permit light to pass through. Second is translucent, where some light passes through but not enough to permit visibility of objects viewed through. The next, opalescent, which appears like an opal stone in color, and iridescent, that exhibits changing rainbow colors when reflected with light. Next is fluorescent, which appears like having one color by reflected light and another by transmitted light. Next is dull, which describes as not glossy in appearance. And lastly, glistening, where it appears like it is glossy when seen. Next. Yan. These are the examples of bacterial cultures, bacterial agar glands, which has the optical characteristic of opaque, translucent, fluorescent, and translucent. Yes, translucent. Next. We have also here some examples of opalescent, dull, iridescent, and shiny. Glossy or shiny. So
Next characterization is the form of growth of the bacteria in a agar gland. First form is the filiform. It has a thread-like structure. Next is beaded. Formation of beads like bacteria attached to each other like a bundle of grapes or other is separated in low proximity. Third is papillate, a form with nipple-like projection. Villus has a cover of villi or leaves like or hair structure. Arborescent, tree-like form. Rhizoid, regular, branched, or root-like appearance. Achinolate, toothed or pointed margins. And spreading, growth, extent, form along the line of inoculation. You will see here the different formation of growth in test tubes of agar gland. We have beaded and arborescent. We have the papillate, villus, spreading, and the filiform. Next characterization is the liquefaction. We have the crateriform. Crateriform is saucer shape, goblet shape, or crater-like appearance. Liquefaction is napiform. Liquefaction with globular top and narrow at the bottom, like a turnip. Next is infundibulum, funnel-shaped structure. Next is saccate, sac-forming liquefaction. And lastly, we have the stratiform, stratified formation extending the walls of the tube and downward horizontally. Here are the examples of the liquefaction. We have the crateriform, napiform of spirillum species, infundibulum, saccate of subculture in gelatin, and we have the stratiform.
So number 7 characteristic is the odor. Microorganisms can produce different types of volatile compounds that may give a particular smell. It may be pleasant scent or pungent odor. The production of these volatile chemicals depends on the metabolic characteristic of that particular organism. Sometimes, scent or odor produced by bacteria can give important clues in the identification of microorganism, but still it is not a reliable rule. We suggest that we should be cautious while sniffing culture plates because sniffing culture plates is not a good idea and you may get a disease. So we have here some example of characteristics of odor of selected microbes. First, we have Alkali genus fecalis, which is, when cultured, smells like freshly cut apples. Candida species, which smells like yeast. Citrobacter species that smells like dirty sneakers. Next is Clostridium difficile that smells like putrid and fecal. We have also another example is Corynebacterium species that has a fruity scent. And lastly, Ichelina, Ichelila corrodens, which smells like bleach or crackers. These are some examples of microbes that have specific odors. Next is the medium change, which give off certain colors as clues to further identify certain traits, behavior, or characteristics of a certain strain of bacteria. When a bacteria is grown at a different agar medium in a different environment. Different culture media contains all the elements that most bacteria need for growth and are not selective, so they are used for general cultivation and maintenance of bacteria kept in laboratory culture. These are some of the examples. First is the Simon citrate agar that contains citric acid as the sole carbon source in the media, and ammonium salt as the sole nitrogen source. This below shows the results of erogenous and E. coli grown on Simon's citrate agar slant. The said media contains the pH indicator bromothymol blue, which is green at neutral pH and blue at pH above 7.6. Next example is the next slide. Next example is the triple sugar iron agar or TSIA. So this test is designed to differentiate different groups or genera of the Enterobacteriaceae. Enterobacteriaceae, a gram-negative bacilli, which is capable of fermenting glucose with the production of acid, and to distinguish them from other gram-negative intestinal bacilli. The differentiation is based on the fermentation of glucose and lactose or sucrose and hydrogen sulfide production, where the acid-base indicator phenol red is indicated by orange red to yellow in the presence of acids. Oxidative decarbolization of peptide that is indicated by an orange red to deep red color, and sodium thiosulfate and ferrous ammonium sulfate are indicated by a black color in the butt of the tube. Lastly, next slide. Lastly is the carbohydrate fermentation test, which uses carbohydrate fermentation broth with Durham tube. The yellow color is indicated by the culture is positive, and the red color with no gas bubble indicates the culture is negative in carbohydrate fermentation test.
Lastly, we have Mr. Sir Gabriel.
What is broth culture? What comes to your mind when you hear the word broth? I'm guessing it's a tasty liquid created from chicken, meat, or vegetables that used as a soup basis. In broth, bacteria and other organisms, such as yeast, can be cultivated. It's not the same broth you get in a soup, but it's close. Broth cultures are liquid cultures that are used in labs to cultivate microorganisms. A scientist starts with sterile liquid growing medium to establish broth culture. The medium is infected with bacteria before put in an incubator at the proper temperature. The broth becomes hazy after given length of time due to increased quantity of bacteria. Why is identification of bacterium's growth patterns in broth important? Identification of bacteria's growth patterns in broth is significant because it allows us to see critical traits that aid in the identification of the bacterium under consideration. How do you determine bacterial growth in broth? Bacterial growth in broth is indicated by the development of a cloudy appearance. If the newly inoculated broth looks cloudy at the start, you will have no way to determine if this is due to bacterial growth during the incubation period. Broth cultures are liquid cultures used in laboratories to grow microorganisms. Before being placed in an incubator at the right temperature, the medium is contaminated with bacteria. Because of, because of the increased quantity of bacteria, the broth gets foggy, and after a certain amount of time, different organisms will exhibit varied growth characteristics in broth. Some organisms will distribute evenly throughout the broth, while others will sink to the bottom and form a sediment, forms, form clumps and create flocculent growth, and float on the top forming a pellicle. Next. Surface growth, none or a ring at upper margin adherent to glass. It's possible that the bacterial strains you're referring to originated from saltwater and bentoic creatures' intestines. Protease production may be responsible for presence of halos surrounding bacterial colonies when they develop in media such as R2A saline medium. B. Pellicle, growth in a form of continuous or uninterrupted sheet over the surface. In broth, different organisms will demonstrate distinct growth properties. Some organisms will disperse equally throughout the broth, while others will sink to the bottom and produce a sediment, develop in clumps generating flocculent growth, and float on the top, making a pellicle. Flocculent, small masses of growth of various shapes floating in a culture fluid. Motile bacteria of those have the ability to swim. Their mobility will cause the broth to become uniformly cloudy. Some of these bacteria congregate in cluster known as flocculent growth. D. Membranous, thin coherent growth like a membrane. Next. This is the example of ring, pellicle, flocculent growth, and membranous. Next. Subsurface growth, none or a, turbid or cloudy. Bacterial growth in broths is characterized by the formation of hazy appearance. If the newly infected soup seems hazy at first, there is no way to tell if this is due to bacterial growth throughout the incubation time. Turbidity is in bacterial solution refers to the optical density of bacterial cells. As bacteria split and proliferate, they occupy the soup, making it appear turbid. When you add something that kills the bacteria, their cells break open and the solution clears up. 2. Granular small particles. For the cultivation and enrichment of non-fastidious bacteria, as well as the evaluation of disinfection performance, peptone and beef extract supplies nitrogen, vitamins, amino acids, and carbon, while sodium chloride maintains osmotic balance. C. Flocculent. Small masses of various shapes. Flocculent growth in broths is best defined as little visible masses of growth, a mass of bacteria at the bottom of a suspension, and visible layer of growth on the surface. D. Flaky, large particles. Flaky just like the granular, but this has larger particles. Next. These are the examples of subsurface growth. Turbid, granular, flocculent, and flaky. Next. Amount of growth. Amount of growth can be none, scanty, moderate, and abundant. Identification of bacteria's growth patterns in broth is significant because it allows us to see critical traits that aid in the identification of the bacterium under consideration. Next. For sediment, non or agranular, small granules. The bacterial swarm near the bottom of the solution, it may contain compounds that restrict development of undesirable bacteria while without hindering the growth of desirable species. Flocculent, small masses of growth of various shapes. Little visible masses of growth, a mass of bacteria at the bottom of a suspension. C. Flaky, separate flakes. Large flakes at the base. D. Viscid, sediment rises as a coherent swirl. Some fermentation systems experience extremely high broth viscosity due to the presence of polymeric or solid substrate, such as in cellulosic material fermentations, the employment of filamentous microorganisms, such as those seen in fungal fermentations, as well as the release of very viscous materials, notably biopolymers. The high viscosity of the soup causes major issues of mixing, heat transmission, and oxygen supply. These issues in turn restrict the fermentation process output, capacities, and efficiency. Next. These are the examples of sediment. 5. Next. Pigmentation. Agrobacterium aurantiacum, Staphylococcus aureus, Chromobacterium violacum, Sriracha mercensis, Bacillus species, Flavobacterium species, and others are capable of creating pigment with various colors such as pink red, golden yellow, purple, red, creamy, and yellow. Next.
In continuation to Mr. Sir Gabriel's presentation, of course, we have certain types of bacteria that may or may not need oxygen to live. Now, what are these types of bacteria? To name a few, we have the aerotolerant anaerobic bacteria, obligate anaerobic bacteria, and aerobic bacteria. We shall now take into account the surface growth as an example for bacteria characterization in broth. As you can see, an example of aerotolerant anaerobic bacteria is the Streptococcus mutans. And regarding its surface growth, it is flocculent. which are evenly dispersed throughout the culture. While they don't require it, oxygen isn't poisonous to them, and they can live anywhere in the culture. Next, we have the obligate anaerobic bacteria. An example of this is the Clostridium species. And regarding its surface growth, it is a sediment. They can survive in the presence of oxygen, so they clump together at the bottom of the culture. Lastly, next, we have the aerobic bacteria. An example of this is the actinomycetes bacteria. And regarding its surface growth, it is a pellicle, which require oxygen to live, form a layer at the top of the medium. This allows them to be as close as possible to the oxygen source. All right, that is all for today. Thank you for listening. And of course, thank you to my colleagues. Have a nice day, everyone.